introduction, and would like to thank the HFSP team for inviting me to share some of our research interests. I'm probably one of the older alumni, got my grant in 2009. I was going to talk about our lab interests. Our lab does a number of things. We are structural bioinformaticians, basically. So on the sequence side, we have been generating computational pipelines so that one can do sensitive sequence searches. We have also developed databases which tries to bridge the gap between the proteins whose structures are available to the number of proteins. There are huge millions of proteins whose sequence alone is available, and one can browse through this at different hierarchies. And also, we've been particularly focusing attention and detailed analysis on some protein families. And on the structural front, we are very interested in the domain architectures, the phenomenon of domain swapping, and the HFSP grant, for instance, was on the myosins, which I will quickly talk about before going to the toll-like receptors. We have done some algorithms that measures the strength of protein-protein interactions that was originally part of the HFSP proposal, which we call as coil check. But this was generalized to all protein-protein interactions, which is called as PP check. And to design mutations to provide a structural rationale for point mutations, these are all mostly collaborative in nature. And what I'm going to talk about on the toll-like receptor is mostly on the virtual screening front, where we recognize effective inhibitors through these approaches. For the past 10-15 years, our lab has been heavily interested in genome analysis, especially transcriptome, genome of medicinal plants, and Ayurveda I'll not be talking about. And it's very tough to take a decision on what topic to discuss. But briefly, on the HFSP proposal, what we attempted to do is to look for all possible myosins in different genomes, which one can then try and understand what sort of interactions are important and what mutations confer hypertrophic cardiomyopathies in the cardiac myosins. And in this context, we had looked at various details, such as how the actin would interact with tropomyosin, which is in turn regulated by myosin, which in turn are also regulated by other calcium-binding proteins, such as troponin C and T. And this gave rise to a huge amount of learning and some publications as well. And the project still continues because recently, for instance, we looked at actin-associated proteins, and even today we are collaborating with University of Warwick on looking at actin-tropomyosin interactions. Just to tell you that and I also thank HFSP for fueling this beautiful project, which we are still very interested in continuing. We would also involving other partners, such as the Professor Jim Spudich, who is interested in the myosins. He is from Stanford. And Henrik Flyberg, who's a mathematician but computational biologist, who helped us with the biophysical data. And these are some of the people in the HFSP project whom we had at that time. So then now going into the toll-like receptor, so these are proteins that are sensitive to the presence of pathogenic materials that come from bacteria or viruses. So just like how the GPCRs will signal the environment, these are they are molecule signaling membrane-bound proteins. And in mice as well as in human, we have a number of them. And some of them will heterodimerize, some of them will homodimerize, and their ligands could be anywhere between lipopolysaccharide to DNA or RNA. And they can sit either in the outer membrane or in the inner membrane. So there's a lot of variety in this, the way the toll-like receptors would interact and recognize and give rise to the signal transduction, which is really downplayed by the TIR domain, whose structure details I'll come to in a moment. But if we focus on one of them, for instance, the toll-like receptor 4, you will find that not only one pathway, but there are multiple pathways by which these membrane-bound proteins can interact. I think many of you already know. I'll go through them rather quickly so that we'll get to the inhibitor design, which is the topic. of today's talk. So the ligand will be interacting on the extracellular region, and the ligand binding causes homodimerization of this receptor, which then is regulated by different levels, because you have adapter domains such as MAL and MyD88, which then gives rise to the signal inflammasome, which eventually gives rise to the NF-kappa B production, which one can appreciate can give rise to the adaptive immunity, which is central for the host to recognize the pathogen attack has happened, and the host is able to take care of itself. But in this process, in the case of toll-like receptors, there's yet another pathway which is mediated by not mediated, but TRAM and TRIF, which are also adapter domains. And perhaps the whole protein, the membrane proteins do get internalized, which is not very, very clear. But there are other proteins such as TRAM and TRIF. And strangely enough, almost all of these adapter domains have this central TIR domain. So it seems to be like a card which is used, the structural card which is used at different layers of regulation, and that is so beautiful. And therefore, we concentrated more on the TIR domains in the intracellular domain, whereas most drug targets have been focusing on the extracellular part of the toll-like receptors. Okay, how does the TIR domain look like? So for that, I would like to present to you the reason why we are interested in the inhibitor design itself, because immune response seems to be important, because that's how the host can take care of the pathogens. But why do we need inhibitors? See, that's what you may be wondering, because at times we also encounter what are called as autoimmune diseases, which means the cell starts to recognize its own small molecules as they are foreign particles, and it can start to be hypersensitive, which we do not want. And an extreme case of the situation could be the sepsis, in which the host starts to slowly kill its own cells. So these are the stages in which the inhibitor design become pertinent. And then I would briefly introduce to you the structure of the TIR domain, which involves a simple globular domain consisting of consecutive beta strands and helices, and beta strands are shown in arrows, and helices are shown as cylinders in this schematic diagram, and this is one of the TIR domain. And when we started the work, there were fewer TIR domains structures available, but you can see that it is pretty globular, and out of all of them, the loop region shown here, which is called as a BB loop in general, is pretty important and a sensitive one, because that's where the dimerization of the TIR domains and the toll-like receptors happen. And in the past, we have looked at how single residue mutations can totally abrogate the signaling at the BB loop, and also other conditions such as phosphorylation can tremendously have an effect. But today, I'll focus on the inhibitor design. So what we can do is to then try and abrogate this interaction, because it naturally happens with a single residue. That means this is a sensitive spot. So most of the time we focus on this BB loop to recognize or design inhibitors, hopefully leading towards drug in the future. And in doing so, we came across in the literature, there are already available peptides, which is called as Viper peptide, which is shown to focus right at the spot which we are interested in. So there is already a naturally available peptide. So our hope is just to do better than that. And starting with the known available information of this Viper peptide, which seems to sit at the BB loop, and also perhaps with the interaction with the adapter domains such as TRAM and TRIF. We wanted to get inspired by the presence of the Viper peptide and its structure, and then come across our own design principles. And for this, we mostly focus on the TRAM region of this signalosome, which means it's the MyD88 independent pathway. And then we focus not only on the Viper site, Viper binding site, also we independently looked at 2 other possible pockets where the inhibitors can bind to. And then we looked into 0.3 million small molecules, which are of natural origin, mostly from plants. And that means we can look at a large number of complex molecules. Many of them have multiple chiral centers and so on. And then we did a virtual screening by which we would focus and reduce the numbers to as many as 3,500 compounds, which go into further screening and molecular dynamics. And why molecular dynamics? Because at a static structure, the small molecule is comfortably binding, but does it stay on or does it fly off? So that's why we do the molecular dynamics, and subsequently we are able to filter as many as 10 compounds starting from 0.3 million, which is quite good, because this is a huge amount of decision making and risk that we take, because we are leaving behind many other molecules which could be equally good or better. But we then went ahead and took an integrated approach by which we would test whether these small molecules indeed stop the signal transduction of this beautiful unit, and using cell-based assays, as well as looking at whether the direct interactions are happening between the protein and the ligands of interest. And I will quickly show you the results. First of all, the cell-based assays were designed such that these are visual spectrophotometric, a little bit like the color visualization we could appreciate in the previous talk. And for this, we construct what is called as a CEAP reporter assay. And if the signal transduction happens, enough kappa B would be produced, right? So because then the gene expression of enough kappa B happens. So if there is no signal transduction, you can see a particular color, which is red. And if there is the substrate, the lipopolysaccharide and the tau-like receptor assembly present, it would be blue in color. So if we do encounter very successful inhibitors, it should look, this color should look red. So the red color means it's a good inhibition, and anything which stays blue means it has not been able to inhibit or stop the pathway. And we can see straight away that in comparison to Viper peptide, which is shown somewhere here, many of the small molecules that we had picked up from our virtual screening, starting from 0.3 million small molecules of plant origin, some of them, like the compounds 2, 4, 9, 10, as we can see from the color becoming red, that means these are successful inhibitors. And side by side, we went back and checked how our virtual screening results had shown in comparison to the Viper peptide, where the interactions happen with some of the TRAM TIR domain. These small molecules 2, 4, and not shown here 9 and 10 are also able to form stable interactions, and these have also been followed through molecular dynamics and shown to be quite stable. So this suggests to us that these could serve as effective inhibitors. And still these interactions are in the micromolar range. However, it outperforms the well-known peptide, which is the Viper peptide. It binds much better than the Viper peptides, as you can see. The smaller, this is the production of the NF-kappa B, basically. So we are currently designing analogs starting from these small molecules. We would basically remove or add chemical groups, and so that we can make them as more effective inhibitors. And to conclude, then the next slide will show you how the NMR results themselves look like. You can see some residues which are shown in blue color are the ones that undergo chemical shifts upon binding to the small molecules. And what I've tried to show here is also the presence, the way the bioinformatics had shown that the ligand could bind, coming from the virtual screening. So the computational modeling of the interaction between the protein and these inhibitors are shown in this green color, and the residues that undergo chemical shifts by NMR is also shown. Basically, it also confers a message that one can look into small molecules from plants, such as the Japanese cornel logboard, magnolia, mango, pomegranate, and other medicinal plants which have already been in practice for combating sepsis. So we can pick up some known molecules, and we can also pick up some unknown molecules, and buy one small molecules from natural origin, because then we'd have to worry less about the toxicity aspects, and the acceptance in the drug market may be faster and more efficient. And all this work was taken by my student, Shailya Verma, who's now a postdoc in the northern part of India. So with this, I will stop and take any questions. Thank you.
Thank you. Thank you very much.